Okay, so chapter 8 is microbial metabolism, which is the chemistry, by chemistry. Um, so the first thing that we need to understand is what are enzymes. This was like kind of AP1, but if we forgot it, generally speaking, what are the enzymes? Enzymes are like the traffic signals. They have specific spots to attach to the substrate. What's a substrate? Substrate is what they are going to work on. Like this is an enzyme that work on the sugar sucrose. So the sucrose is considered the substrate for the enzyme sucrase, for example. Lactose, lactase. So lactase is an enzyme that work on lactose. What's lactose? This is the substrate. Okay? Each enzyme will have an active site in which the substrate is going to attach. Of course, if you look at this picture, you see the enzyme, the big green part. Look at the substrate and look at this active site. You will see they look the same. Look at this and look at this. They look the same, so they can fit in each other. And this is how the enzyme know that this is the specific substrate that's supposed to work on. If it can fit into the active site, you can do it. There is another site usually that's called the regulatory site. There might be or might not be a regulatory compound that can fit in here to either allow or not allow. The metabolic pathways. Metabolism, generally speaking, when you say metabolism, you mean two cycles or two circles, catabolism and anabolism. You build and break continuously. You call that metabolism. Okay? You get something, you eat it, you break it down, and then you build. You break it down and build. So it's an alternation between anabolism and catabolism. Ana means building, cata means breaking. So anabolism means the building, you build something. Catabolism means you break down something. So when you say anabolism means you get small pieces, you put them together to make a big component. Like when you get the bricks, you put them together to make a wall, right? We call that anabolism. The opposite is, give me the wall, I'll break it down into bricks, and I call that catabolism. All right? Source of energy, it depends on which organism are we talking about. I would say that the ultimate source of everything in life is sunlight, right? Like if you think about yourself, how do I get the energy from? I get the energy from breaking down the organic molecules, right? I get carbohydrates, fats, proteins, I break it down, especially carbohydrates, and I get my energy out of it, right? Where did you get that from? From animals and plants. Where the animals got it from? From the plants. Where the plants got it from? From the sun, right? So this is the ultimate source. If there is no sun, no plants, no animals, no humans. So this is the very, very beginning. This is the main source that will go from organism to organism to organism. But the ultimate source, the primary source, is obviously sunlight. So when we say catabolic pathway and anabolic pathway, catabolic means to break down. And when you say break down, you mean to break it down to release energy out of it. Okay? So it goes like this. Look at this here. If I have these, this is a compound like this, right? Here is a compound. When I break down, I release the energy and I use it. Now when I have extra energy, I'll get this one, put some energy, put the next one. Put some energy here and put the next one. So now when I do anabolism, I'm storing energy. When you break down, you release energy. Is that clear so far? Anabolism, using energy to build. Catabolism, breaking down to produce energy. So it's the opposite of each other, but it always happens in cycles. So again, when you say metabolism, what do you mean? I mean all these chemical and physical working of the cells, which is catabolism and anabolism, okay? So what's metabolism? It's an alternation of anabolism and catabolism. Both of them together, I call it the metabolism. Catabolism is you break down something, you degrade it to get the energy out of it, right? A big compound, you break it down into smaller pieces and you get the energy out of it. Anabolism is exactly the opposite. Anabolism means synthesis. We call it biosynthesis, which is formation, building. Biosynthesis means building. You make something larger from small molecules, and obviously that's going to require energy. Is that clear so far? Okay. So when we say enzymes, this is like a light signal that's going to have two very important criteria that you need to remember. Okay? Why do we have the enzymes? Number one, lower the energy of activation. This is one. The other thing is to speed up the biological reactions. Okay? And they work on specific substrate. Let me just give you an example to make it easy for you, for the enzymes. Let's say if I have my car and I have a flat tire, right? I have a flat tire in the street and I need to replace it. If I don't have enough tools, if I don't have good tools, I'm going to use whatever I have, right? I have this wrench and I'm going to use like a piece of rock and put it up and try to take it out. That will take time and effort, right? Time and effort. What if I have like this hydraulic machine? It will take me few minutes, right? So less time and less energy, right? This is exactly what the enzymes are doing, just like a hydraulic machine. It makes the reactions faster and use less energy. Like if you have the machine, how long does it take from you to replace the tire? Maybe 5-10 minutes, right? Because you have the machine that's helping you. How much energy? You're not using much energy. You're not using your physical energy. You have a machine, right? So the two important thing about enzymes to remember is lower the energy, speed up. Lower energy and speed up the reaction. And you need to remember that they are very specific because of the active site. Okay? Each enzyme will work on a specific substrate. The enzyme is protein in nature. Okay? And these simple enzymes are just proteins. That's it. These are the simple ones. But there are a little bit other types of enzymes that are a little bit more complicated, and we call that the conjugated enzymes or holo enzymes. Holo, conjugated, holo, whole, big. This is a protein and another molecule that's not a protein. The protein part, we call it the apoenzyme. The other part that's not protein, we call it a cofactor. The cofactor is inorganic element. Inorganic elements. Everything that I'm talking about is important, okay? I'm focusing on the most important parts. But the vast majority, basically, but if something that's not very important, I will not focus much on it. So far, the enzymes can be simple or conjugated. Is that okay? Simple, conjugated. If it is conjugated, you also call it holoenzymes. You need to remember both names. Conjugated is the same as holoenzymes. What do you mean conjugated or holoenzymes? It means it's not simple. It's not just a protein. There is another part. What is that part? It's the cofactor, which is inorganic elements. Usually it's metal, like iron, copper, magnesium, something like that. So for the enzyme to work, you need that cofactor, okay? Sometimes you can need organic molecules, but these are called coenzymes. It's not the regular cofactors. Is that okay so far? Cofactors are not the same as coenzymes. Cofactors, if it is inorganic elements, that's one thing. If it is organic, we don't call it cofactor, we call it coenzyme. Coenzyme is usually a vitamin. Okay? Coenzyme is organic, vitamin. It's organic and it's usually vitamin. Cofactor is usually inorganic, metal, most likely. Metal ions like iron, copper, magnesium. So if you look at this purple part, what do you call this whole thing here? The purple and orange. Hollow enzyme, or conjugated, the whole thing, right? This part of it is called, this is the apoenzyme, right? And this part is the cofactor, right? If you put it together, that's the hollow enzyme. This is if it is conjugated or hollow enzyme, okay? Some of these will need coenzyme that will, beside the cofactor, some of them will need the coenzyme, which is organic, and most likely it's vitamins. And this is what we usually need. So do we need cofactors or coenzymes? Both of them. Some enzymes need cofactors, some enzymes need both of them, right? So if you ask yourself, why I'm eating vitamins, is that something that to get energy out of it? No, it's not to get energy out of it. It's to help the enzymes to work. It's an organic molecule that helps the enzyme to work. Okay? The enzyme needed. These are catalytic enzymes or catalytic reactions. And you don't have to memorize all of this. Just understand the idea. This is for you to understand. Okay? So if you look at catalase, for example, let me tell you this. Generally speaking, any enzyme should end with, or most of them, should end with ASE. Okay? So if you have something like lactose, what's the enzyme that work on lactose? Lactase, lipid, lipase, protein, protease. So it's the same word, just change the end and make it ASE. So these are all catalytic action, or these are all reactions that the enzyme help, like catalase is helping a reaction, and the cofactor is iron. What if I don't have enough, or the ferrous? What if I don't have enough iron? Is the iron cofactor or coenzyme? Cofactor. Why is it cofactor? What's not coenzyme? It's metallic, inorganic, exactly. What if it is a vitamin or organic? You call it? Coenzyme. Great. So this is what the cofactors or coenzymes needed. This is not all just metallic cofactors. Some of these are coenzymes. Like thiamine is a vitamin, for example. Thiamine is one of the vitamins, so it's considered coenzyme. Okay? So cofactor, anything that helps the enzyme. Inorganic, this is just a cofactor. If it is organic, you call it coenzyme. Okay? And these are just examples. This is too specific, so you don't really have to remember everything here. Just look at it to understand how it goes. Okay? And if you noticed here, those on top, everything here is just a cofactor. Look at these. These are all metals, right? Iron, copper, magnesium, manganese, zinc. These are all metals. So what do you call all of those? 1, 2, 3, 4, 5, 6, 7. Cofactors. These two, thiamine and fat, we will talk about those a little bit more. But these are vitamins. So what do you call it? Coenzymes. The active site is called the catalytic site. And the active site is, this is a site where the substrate is going to bind to. And I mentioned that it looks like the substrate, right? Or the part of the substrate that fits into this. If it fits in, the enzyme will work on it. If it does not fit in, the enzyme have nothing to do with it. The enzyme is always very specific. Okay? So substrate attached to the active site in the presence of cofactor at the active site. You do the reaction that's needed with the help of the enzyme, and then you release the product. The idea here is, remember that the enzyme usually does not change. Okay? Like if you're using the hydraulic machine, after you're done, is the hydraulic machine is done? Is it gone? Is it over? Or you still have it? Right? You still have it, and you can use it as many times as you need to. So it's not consumed in the energy. Is that okay? It does not change. It's still available. Use it. Reaction is done. You still have it. Use it again, over and over and over again. It does not. It's not consumed and does not change. So if you look at this, this is the substrate. Look at those two substrates. Which one will fit? This or this? Obviously this. So the enzyme is specific in this substrate S, right? Can we work on this? No, it doesn't fit. Another enzyme can do it, but not this one. Okay? So it fits in, and then you get the reaction, and then you get the products. So the active site is the catalytic site, and this is where the substrate is going to bind. The enzyme will be unchanged. And this reaction, you see that did the red or the orange purple is the one that can fit. Reaction is done, and then you have the substrate released. So here it is again, just reinforcing the concept. What's the difference between the metallic cofactor and the coenzyme? Metallic cofactor is metal, it's inorganic. The coenzyme is organic. The metallic cofactor will activate the enzymes. This goes to the active site so that the substrate can bind to. While the coenzyme work as a carrier to the substrate. It carries, it's vitamins. Most electron carriers are coenzymes. We will get to that point later on, but generally speaking, you need to remember that later on, when we do the reactions, you will see that you are going to need to transfer and to carry electrons from one spot to the other. Who is going to carry that for you? It's coenzymes, which is vitamins. Did you see that there is a difference between those two? Not just in the nature, in the function. Is that clear? What's the difference in the function? The composition is different. It's different parts. This is metal and this is vitamins, right? This is inorganic and this is organic. Okay, but what is the function? It's different. The cofactor, the function is to activate the enzyme by going to the active site, allowing the substrate to attach. Is that okay? This is for the cofactor. The coenzyme is playing a different role. It's a carrier. It carries the substrate to help it to fit in. Like electron carriers, coenzymes also work as electron carriers. So it's carrier. Carrier for what? For substrate. What else they can carry? They can carry electrons. So what the one word that you have to remember about the cofactor? Carrier. Carrier. Carrier of what? Carrier of substrate, if we're talking about these enzymes, or carrier of electron. We will come to that point later on. Now, depending on the location of the enzyme, we are going to divide the enzymes into, it's very simple. It's endo and exo. Endo means inside and exo means outside. Is this reaction helping reactions inside of the cell or outside? If it is helping reactions inside, if it is all the time inside, intracellular, we call it endo enzymes. If this enzyme is working outside of the cell, we call it exo. Exo means outside. Like amylase, cellulase, what are these enzymes? These are enzymes that go out of the cell, it's released out, and they work outside of the cells. So these are the exo enzymes. See how they are working outside? The purple ones. They are released out. You kick it out first, and then it will work outside of the cell. We call it exo enzymes. While these yellow ones are the endo enzymes. They work inside. Another differentiation of the enzyme is constitutive versus regulated enzymes. Constitutive enzymes is always present. uh, and they are produced in equal amount or equal rate regardless of the amount of the substrate. And this is something unique to remember. Constitutive enzymes. These enzymes are not produced exactly because of the substrate. You produce it no matter what. Did you get the idea? You make it anyway and keep it. You have a lot of substrate, you have a little bit of substrate, it doesn't matter. I'm going to make it anyway and keep it anyway. Is that clear? It's usually inside of the cells, but it's usually inside. But should I call it endoenzymes? No, I call it constitutive. What's the difference? Constitutive are different than endoenzymes. Endoenzymes, they work according to the substrate. I have 5 substrates, I use 5 enzymes, okay? I produce and use 5 enzymes. While the constitutive enzymes are different. We produce it irrespective of anything. I don't care about how much substrate, I'm going to produce it anyway. Okay, you got this point? Regulated enzymes are the enzymes that are not always there. You produce it as needed. So constitutive, do you produce it as needed? No, you always produce it. You produce it and keep it. Put it in the side. If you produce it as needed, you call it regulated enzymes. So these regulated enzymes, what do you mean regulated? I mean these enzymes that I can regulate by using an inducer or repressor. If I wanted to use the enzymes, I induce them. I add something to it to induce it. If I don't want to use it, it will not be available, it will be repressed. So does it change because of the substrate concentration? Yes. Is that clear so far? So depending on the rate of production of the enzymes, it's either constitutive or regulated. Constitutive, always there, always produced, irrespective of the substrate. Regulated means as needed. And you use the inducer and the suppressor. You need it, use the inducer. You don't need it, use the suppressor. The enzymes are classified according to their function into different types of enzymes, depending on site of action, type of action, what substrate. So this is going to give us a different classes of enzymes. And most of the name come from changing the last part of the name into ACE, A-S-E, right? Lactose, lactase, lipid, lipase, protein, protease, okay, and so on. So example here is lactase, and it works on lactose. And the class is hydrolase. DNA polymerase, it obviously work on DNA, right? DNA polymerase, it work on the DNA polymer, which is that DNA nucleotide. But this type is called synthase. Other types are oxidoreductase. It does oxidation and reduction, and we will talk about those. But generally speaking, here is what I want you to know. In all this table, here is what I want you to know. Number 1, they will end with ASE. And the other part of the name is the name of the substrate. Is this part okay? This is number 1. Number 2, hydrolase versus synthase versus oxidoreductase. What are these 3 different classes? Hydrolase means you are going to use water to break down. So is that anabolic or catabolic? Catabolic, because you're using water to break down. Isn't that catabolic? Yes. Synthase is the opposite. This is an anabolic reaction. So synthase is you get different small components and you put them together and you make something. You end up making something. So hydrolase is catabolic. Synthase is anabolic. Hydrolase you're using water to break down. Synthase, you produce water. Oxidoreductase is you oxidize something and you reduce something else, okay, by adding or removing electrons, and this will come later. So dehydration and hydrolysis. I showed you 2 examples, right? Synthase, I showed you actually 3, but I'm talking about the first 2. I said hydrolase and synthase, right? Hydrolase and synthase. Hydrolase, the reaction that you do hydrolysis. Synthase is when you do dehydration synthesis. Okay? Here is simply what it means. Let's say this is anabolic reaction. I will show you. Look at this. Let's say you are glucose, okay? And I am another glucose. We need to react together and make a disaccharide. We need to come together. If I am glucose and she's glucose, we need to come together. It's not going to work this way. The only way to force us to interact with each other is to take hydrogen from one and OH from the other one. That would be water, right? Dehydration. Did you take water out of both of us? You take hydrogen from me, you take OH from her, so now you have hydrogen, OH. Isn't that water? Dehydration. You removed water. Now, what I'm missing hydrogen, right? What she is missing, she's missing OH. She cannot wait, she cannot stay without the OH. I cannot wait without the hydrogen, so we come together and make the disaccharide. I call that dehydration synthesis. Is that okay? Now we are together. We are one molecule of disaccharide. Can we split? You cannot split. She will say, okay, give me my OH and then I can split. And I will say, give me my hydrogen and I'm going to split. So you're going to add the water. When you give the water, she takes the OH, I take my hydrogen, we can split. So now you added water, right? Hydrolysis. Did you understand the concept? So dehydration synthesis versus hydrolysis. Which one is catabolic? Dehydration synthesis or hydrolysis? Hydrolysis is catabolic. And what hydrolysis means? You split by adding water. Dehydration synthesis, is that anabolic or catabolic? Anabolic. And how do you do it? Take out water. Dehydration. Remove water. Clear so far? So this is dehydrolysis and dehydration. And can you answer this? Concept check. Which one is right? C. They may be active extracellularly. Is A correct? No, they are specific. Are these supposed to be consumed in reaction? Never. They always composed of protein? It can be what or what? It can be protein alone or protein and non-protein, which you call it holoenzyme or conjugated. Yes. Now the enzymes are sensitive to the environment. What is the nature of the enzymes? What's the basic part of the enzyme? Is it carbohydrate, fat, or proteins? It's proteins, right? So being protein, the protein can change the nature depending on the temperature, pH, and osmotic pressure. So if any one of those changed, something can happen to the enzymes, right? It will be denatured. So if you change the environment, so for the enzyme to work, you need to have a perfect environment. What do you mean by perfect? I mean perfect temperature, perfect pH, and perfect osmotic pressure. These enzymes are going to work just fine. Okay? If these enzymes are chemically unstable, we call it labile enzymes. If the bonds between the components of that protein are weak and can be broken, you call that denaturation. I'll give you a very simple example. What do you think? Here is an egg. I get an egg, a raw egg, okay? And you open it. How does the white of the egg looks like? Is it really white? It's not white. It's clear, isn't it? Transparent. This is the nature, right? What happens if you expose that to heat? It becomes white. Isn't that denaturation? Means you change the nature from clear fluid into a solid white, isn't it? Two changes. You're changing the color and you change the liquid into solid. Isn't that the case? I call that denaturation. Let me tell you something. We all know that this is because of the temperature, right? If you get some of it and just expose it to the heat a little bit, that's what's going to happen. Let me tell you something here. Even if I get some acid and I put it on the eggs, I'm telling you, the same change will happen. If I get some oven cleaner and put it on the egg, same change will happen. It will be denatured. So what's denatured? Changing the nature. Why? If these, like in this case, the egg, the bond is very weak. So just expose it to a little bit heat, it will be denatured. Change the pH a little bit, as in adding acid or base, it will denature. Okay? The metabolic pathways are basically these group of reactions that go from step to step to step to step as you need it. It's not as simple as I just mentioned, like lactose with lactase. It will split into galactose and glucose. It's not that simple. If let's say another reaction, here I have glucose, right? And I'm going to break it down. It's not one step, right? I'm going to, here I get glucose, cells are going to use it, and it will become water and carbon dioxide. It's not that simple. You have to go step after step after step after step until you get to that point, okay? The very important regulation here is the feedback. And here's what I mean by feedback. Are we familiar with glycogen? What's glycogen? Is that a polysaccharide, disaccharide, or monosaccharide? It's polysaccharides. Polysaccharide of what? Glucose, right? It's multiple glucose units. So if I break that down into these glucose units, right? And breaking down, I get more glucose. Breaking down, I get more glucose. At certain time, you have a lot of glucose. And then you are going to say to this, stop. You call that negative feedback. And this is the most important regulation. Accumulation of the end product will lead to slowing down of this type of reaction. Did you get the idea? If you're breaking down a lot of glucose and you're accumulating a lot of carbon dioxide, the reactions are going to slow down. Who slowed down the reaction? Negative feedback. This is the most important regulation, and it depends on the amount of product. Okay? If you are producing something that's something good and you need it, right? But you cannot overdo it. You cannot make too much of that end product. Now the action of the enzymes are controlled by different way. Okay, one of them that I mentioned is the negative feedback. Negative feedback is going to slow down the enzyme. This is one. Now, how are you going to slow down any enzyme? There are two ways to slow down an enzyme: the competitive way and the non-competitive way. The competitive way is two enzymes compete on the same substrate for the active site. Okay? If this is something that resembles another thing, they are going to compete on that for that active site. So this is a competitor inhibition. You are going to inhibit the enzyme by competing with that enzyme. The non-competitive way of inhibiting is that you inhibit by inhibiting the whole enzyme substrate complex. And that's why we will have an enzyme repression and enzyme induction that we talked about before. You can remember these regulated enzymes? Do you remember this? We said there is a constitutive and regulated ones, right? Constitutive, you just make it, irrespective. But the regulated ones, you make it as needed. You activate them as needed. How to do that? Induction versus repression. Is that clear? I have 10 units of lactose and I need 10 units of lactase enzyme, right? How to do that? Induce 10 and repress the rest of it. If you're eating more, 20, okay, I'm going to induce 20 and repress the other. So you can induce or repress the enzyme as needed. This is going to be repression versus induction, and this is not competitive way. So you're not competing with the enzyme. You're actually, here is exactly what's happening. Here is an enzyme, okay, and it's fitting into the substrate. You are not going to compete with him. Just leave him alone, okay? So you are working on this, that's fine, work. Another one is working, that's fine. And then I'm come to attach to you and stop you. I'm not competing with you. Did you understand the idea? So the non-competitive way is you're not competing, you leave it alone. And while doing the work, you come and repress it. You repress what? You repress the whole complex, not just the enzyme. Look at this picture, that will extend it more. This is competitive. This is the substrate and this is the enzyme, all right? Normally what's going to happen? Substrate fit into the enzyme. Okay. If you don't want that to happen, why you don't want that to happen? Because you're overproducing. It's too much. You get this competitive that fits into that spot, not allowing the normal substrate to work. Did you compete on the active site? Did you get the idea? This is competitive. But in this way, look at this enzyme substrate, leave it alone. It fits in, leave it alone. And then you add the suppressor in this case. You add the suppressor after the complex is formed. So here you do not allow the enzyme substrate to form in the first place, right? By competing. Here, fit in, interact, you're done, okay, I'm going to stop you. Did you get the idea for this? Generally speaking, the energy is defined as the capacity to do work or do a change, cause a change. And energy can be thermal, can be electrical, mechanical, and so on. But the one that we are actually care about is the ATP type of energy. This is the most important one. When we talk about any reactions, some reactions will need energy, will consume energy to do the reaction. We call that endoganic reactions. Some reactions are actually going to produce energy. I call that exergonic reaction. What's endo and what's exo? Endo is in and exo is out, right? So endoganic means you put energy in. Clear? You put energy in the reaction to make the reaction goes. While the exergonic means you produce energy out of the reaction, okay? And which one do we have? We have both of them, okay? And these are going to work together. Here is, let's say, look at this example for example. Look at this. I'm going to have X and Y I don't need energy, I actually produce energy. What do you call this? Exergonic. So now I have energy, right? I got it. I'm going to use it for another reaction that's endergonic, so that this reaction will move on. Okay? So exergonic versus endergonic. Okay, can you tell me which one takes energy to work? Exo or endo? Endo. And which one produce energy? Exergonic. Okay. Now when we say redox reaction, redox reaction is, it's split it into two parts, red and ox. Reduction and oxidation, they always work together. You reduce something in order to oxidize something. And we mean by that is electron donor and electron acceptor. Electron donor is the one that's going to be reduction, and electron acceptor is oxidation. Okay, and we will come to that. If you look at this example here, you don't need to memorize these formulas or anything, but just look at it. You see C12H2O6, right? The ratio is 1 to 2 to 1. Did you notice this? You don't need to memorize specifics. Just this is generally speaking 6 carbons, 12 hydrogen, and 6 oxygen. This is a monosaccharide. Okay? This is carbohydrate, 1 to 2 to 1. So this is glucose. And you're going to oxidize glucose. How are you going to oxidize? You add oxygen to it. The oxygen will be reduced and the glucose are going to be oxidized. One more time. Here is glucose. I need to break down this glucose. Meaning, I need to oxidize it. I take something out of it. How do you do that? By using oxygen. The oxygen is going to take it. So the oxygen is going to be reduced. So you're oxidizing glucose with oxygen. Oxygen will take it, so the oxygen is reduced. So you end up oxidizing glucose and reducing oxygen. So it usually works in a pair like this. Are we clear here? So you're oxidizing what? You're oxidizing glucose. You break it down and take something out of it. Clear? When you take something out of it, you're oxidizing it. Where is that something going to go? To something else. That something else that take it, you call it reduced. You reduced it. Why are you doing this oxidation reduction reaction? In order to produce energy. We have a lot of details to talk about this, but generally speaking, so far, glucose will be oxidized. By adding oxygen, the oxygen will be reduced to produce energy. Glucose, break it down, you end up having the end products, and this is a redox reaction. Now, what's actually going to happen here is that you're going to work on the electrons of this substrate. Like in this substrate, we're talking about glucose. Glucose is C6H12O6, right? So you have these carbon molecules, hydrogen molecules, and oxygen molecules. Obviously, these are all atoms, right? Isn't hydrogen is an atom? You're going to take the electrons out of it. You're oxidizing it, right? And you're going to take it somewhere else. Who is going to carry these electrons? If you are breaking down glucose and you're taking electrons out of it, who is going to carry it for you? It is the cofactors. Do you remember that slide? What do you have to remember about coenzymes, I mean? Carriers. Carriers. Coenzymes, carriers. Carriers of what? Carriers of the substrate or carriers of electrons. Clear? So who is going to carry The electrons for you, the carriers are coenzymes, which is vitamins. Is that organic or inorganic? Organic. So what are these coenzymes, which is vitamins, that we will need to learn? NAD and FAD are the most important one. NAD, you don't have to know the details, but generally speaking, NAD means niacin, adenine dinucleotide, and FAD is flavine, adenine dinucleotide. Irrespective. Niacin and flavin, the first, let's just remember this one only. This one letter here, and this. This is niacin and this is flavin. These are vitamins. Okay? So what's the NAD and FAD? These are carriers that come from vitamins. NAD come from niacin, FAD come from flavin. What's the niacin and flavin? Vitamins. Isn't the vitamins are coenzymes, carriers? They're going to carry the electrons for you. Okay? Carry it from the glucose when you break it down and send it somewhere else. Where are you going to take it to? You're carrying these electrons. Where are you taking it? To oxidize something else. To reduce something else. If this reaction is aerobic, you're going to reduce oxygen. Is that clear or not? If it's not clear, just tell me. Okay? Is that clear? Yes. Let's say this is glucose. We have glucose here, okay? 1, 2, 3, 4, 5, 6. C carbons, and it's irrespective. I'm not going to ask you about this, okay? So we have some hydrogen and some oxygen here, right? And I'm going to break down this, okay? And I'm going to take something out of it. So this will be oxidized. Clear so far? Okay. What are you going to take out of this? I'm going to break it down, and I will take the electrons away. Who is going to take the electrons away? NAD and FAD. What's NAD and what's FAD? Coenzymes, right? So the coenzymes. When you break this down, I'm going to take the electrons out of it. Who is going to take the electrons? Who is going to carry it? NAD and FAD. What's NAD and FAD? Coenzymes. Vitamins. They come from vitamins. NAD is not a vitamin. It came from vitamin. Niacin is vitamin and flavin is vitamin. Are we clear so far? So you break it down, and then electrons, you're going to take it from there. So I take the electrons. I'm NAD or FAD. I take. Where are you taking those? To anything that will be oxidized. If this is aerobic, I'm going to take it to oxygen. Okay? So I will take the electron from here all the way to the oxygen. And when you take the electron, you take the hydrogen with it. Okay? Hydrogen. Remember hydrogen that have a proton here and electron out here? So you take the electron and the hydrogen. I take both of them. So when I take it from the glucose, after I break it down, I send it to oxygen. Oxygen will become what? You're taking the electron and hydrogen here. What happens when you add hydrogen to oxygen? Water. Yes, water. Did we get this point? It is glucose. This is my substrate. I need to break it down and get energy out of it. Okay, break it down. Now I will take the electron and hydrogen. Who's going to carry it? The carriers. FAD and NAD. Where are you taking it to? I will take it to oxygen if available. What if it's not available? I can use something else. It's coming. But did you understand this so far? Okay. If this is an aerobic reaction, what do you take it to? What's aerobic means? You have a lot of oxygen. Oxygen is available. Anaerobic means you don't have enough oxygen. So if this is aerobic, you take it from this into oxygen. This is aerobic, right? What if that's anaerobic? You still take it, right? But not to oxygen, something else. As long as it's something else, irrespective of what it is. You take it to something else, this is anaerobic. If anything not clear as we go, just let me know, okay? So can you answer this? When you say something reduced, what do you mean? Did you lose or gain? Did you lose or gain first? Gain. Gain what? Oxygen or electron and hydrogen? Like this. What did you lose? What did you lose here? Hydrogen, right? And what else? I said, look at this. You have electron and the rest of the hydrogen, right? You take electron and hydrogen and you put it to something else. This is oxidized and this is reduced. They are asking about reduce. Did you gain or lose? Gain. Gain what? Yes. Did you get it or not? Okay. When you talk about energy, the most important to talk about is adenosine triphosphate or ATP. This is, we call it the metabolic currency. This is like the dollar, right? You take the dollar and you spend the dollar, right? You save the dollar, you spend the dollar. This is exactly the ATP. And ATP consists of 3 parts. Adenine, which is a nitrogenous base. Ribose, this is a 5-carbon sugar. And 3 phosphates. And here's what's going to happen. So look at this, just to get the idea. This is adenine. Okay? Look at this. This is adenine. I'm going to add sugar to it. Okay? Now I have adenosine. When you say sin at the end, it means you added sugar to it. S-I-N. Clear? What is this? Adenine. I'm adding sugar. 5-carbon sugar. What do I have now? Adenosine. Adenine become adenosine. Now look at this. I'm going to put some energy and add 1 phosphate. Energy, add phosphate. Energy, add phosphate. You see this? Adenine. Now it's adenosine, right? Put energy here, phosphate. Put energy here, phosphate. Put energy here, phosphate. So what do you call all of this? Adenosine triphosphate, isn't it? Now did I store now or release energy? Stored it. What if I need it? Look at this. I'm going to break down 1 phosphate. You're releasing the energy that was here. So what do I have now? What is this and what is this? ADP, and this is phosphate. One more time. Look at this. Adding. Okay? I will add sugar to it, which is 5-carbon sugar. What do I have now? Adenosine. I have plenty of energy. I'm going to put some energy here and 1 phosphate. Did I store the energy here? Yes. I still have some more energy. Put it here. Add 1 phosphate. Still have more energy. Put it here. Add 1 more phosphate. I have ATP now. Okay? ATP. Do you see 3 phosphates? 3. I need the energy. Now release it. Break down. I have 1 phosphate and ADP. And I release the energy. Okay? So why don't you break it down more? No, this is how we use it actually. Okay? Well, what if I have extra energy? Put the energy here, add the 3rd phosphate. I have ATP now, okay? I don't have energy, give me more. Give me some energy. Break down, I have ADP, phosphate, and I release energy. Now I'm eating, and I have a lot of energy. Store it again. Put it, and return this. Return the 3rd one. So I always alternate between ADP and ATP, okay? This is exactly how it goes. Always. You eat, you gain energy, you make ATP. You didn't eat, you need your energy, you break down ADP. So how about, can we break the ADP not for the purpose of energy? We use it for something else, which is not our issue. We can use the ADP, we can break it down into AMP, which is adenosine monophosphate, but this is not for the purpose of utilizing energy. If you only need energy and that's it, you break down ATP into ADP and phosphate. Is that clear so far? Okay, so look at this picture. Here is adenine, and this is the five carbon sugar. What do you call those together? Adeno sen. Adeno sen. When you add one phosphate, what do you call it? Adenosine monophosphate. You add another one, adenosine diphosphate. You add the third one, triphosphate. Okay? Now how you gain and use, or what exactly do you do to gain, and what do you use it for? I gain by respiration, and this is the one that we're going to cover more, or you can get it by fermentation, photosynthesis, chemoautotrophy. Any one of those will give you energy. Okay? And how? What's the use for that energy? You're going to use it for different things. Chemical work, you can use it for mechanical work, transport, and so on. Okay? You gain from one of the four, and you use it for any one of these works. The ATP synthesis. When you add phosphate to the ADP, what do you get? I have ADP and I add phosphate. You get what? ATP. Adding phosphate is called phosphorylation. You phosphorylate something. You add phosphate to it. Okay? Oxidative phosphorylation or photophosphorylation. What's the difference? Both of them are phosphorylation. What does it mean? What phosphorylation mean? Adding phosphate to the ADP, right? In order to add it, I need energy. Depending on where did you get the energy from? Yes?
So when your body is using it in the form of ADP, where does the extra phosphate go?
You just keep it in the cells.
Okay.
Like I have the phosphate in the cell, I have the ADP in the cell. Both of them stay.
It only attaches when you need the energy.
Exactly. When you have energy,
Right.
you attach if you have energy, and you break down if you need the energy. Okay? When you add it, you call that phosphorylation, right? But remember, before you add it, I need energy, right? So where did you get that energy from? But what do you call it first? This is ADP, and I add phosphate. What do you call this? ATP. And what do you call this process? Phosphorylation. Phosphorylation means to add phosphate. You phosphorylate something. Is that clear? Phosphorylation. If you get that energy from oxidizing something like glucose here, I call that oxidative phosphorylation. On the other hand, like plants, for example. Plants, they do not get glucose from something and break it down. Of course not. They get it from sunlight, right? So we call that photophosphorylation. So in both cases, you are phosphorylating anyway, right? Phosphorylating using what? It depends. If you're using organic molecule or you're using the sun. Now, bioenergetics is studying everything about energy, the mechanism and everything else. Here is exactly some details. Remember I told you this, right? You get the glucose into the cells, you break it down, and you take electrons and hydrogen away, right? Through the carriers. But in order to do that, it's not that simple. Okay? So what's going to happen exactly if you have glucose? I have 3 cycles that we need to learn. The first step will be glycolysis. Glyco means glucose. Lysis means breakdown. So glycolysis is to break down glucose. How many carbon molecules in glucose? 6 carbons, right? All that's going to happen in glycolysis is, this is a 6 carbon molecule, I will split it into 3 and 3. Right? It is a 6, I split it into 3 and 3. Those 3 and 3 are called pyruvic acids. So that's it. Glycolysis. What's glycolysis? To break down glucose into 2 3 carbon molecules. This 3 carbon molecules is called pyruvic acid. Is that clear so far? And this step is going to happen in the cytoplasm. Yes, everything is important that I'm talking about. If this is not important, I will tell you. It is important. What's glycolysis? Break down glucose into what? 3 carbon molecules called pyruvic acid. Where is that glycolysis happening? In the cytoplasm. In the cytoplasm. Okay? And that so far does not need oxygen to work. It does not need oxygen. So we call that the anaerobic part. Glycolysis is the anaerobic part. Now, if you have oxygen, you are going to take that pyruvic acid into the mitochondria and to do the other 2 reactions, Krebs cycle and electron transport chain. So if you can do only glycolysis, you end up having 2 pyruvic acid, right? Is that clear? I have 1 glucose, I break it down into 2 pyruvic acids. The 1 glucose is 6, the 2 pyruvic acid is 3 each, right? You break down into 2 pyruvic acids. Is that clear? Where is that happening? In a cytoplasm. Did I need oxygen? No. You didn't need oxygen yet. What if you don't have oxygen? That's it. I do only glycolysis and stop. So we call that the anaerobic part. Clear so far? What if I have oxygen? I call it aerobic, right? So what are you going to do? You will say, and this is normally what's happening. You will say, okay, I broke down glucose into pyruvic acid. Pyruvic acid still have plenty of energy. I don't throw it away. I can still neutralize it. So we're going to Krebs cycle, also known as TCA, or tricarboxylic acid cycle, or citric acid cycle. You need to remember the 3 names. Krebs cycle, tricarboxylic acid cycle, citric acid cycle. It's the same thing. And the last one is electron transport chain. So, can you tell me, if you're following, step number 1 only, glycolysis, is that aerobic or anaerobic? Anaerobic. What if it's glycolysis? Step number 1, plus 2, plus 3, all of them. Is that aerobic or anaerobic? Aerobic. Glycolysis alone, anaerobic. All 3, aerobic. Because glycolysis will happen anyway. I don't care if you have oxygen or not. The only difference is, after glycolysis, if you have oxygen, you do 2 more cycles. You don't have glycolysis alone. So glycolysis alone is anaerobic. Glycolysis, Krebs, electron transport, all 3 aerobic. Here's the question. Why do you have to have oxygen in order to do the other 2? Because those 2 are going to happen in the mitochondria, and the mitochondria will not work without oxygen. Okay? The mitochondria will not work without oxygen. So you have 1 reaction only or all 3. These are the 2 alternatives. So aerobic is glycolysis only. Anaerobic, glycolysis followed by Krebs cycle followed by ATC. Anaerobic is glycolysis only. Okay? Fermentation is a form of anaerobic, and this is, you're not using oxygen as your end product. So here, the acceptor here is not oxygen. You call it fermentation, meaning you're doing reactions without oxygen. So what's going to happen to the pyruvic acid? Here is the question. If this is aerobic, the mitochondria can work, right? If you have oxygen, mitochondria will work. Mitochondria can do Krebs cycle and ETC. Is that clear? So if I do have oxygen, all 3 will happen, right? Correct? What if I don't have oxygen? The mitochondria will say, no, I'm not going to work, right? So now we have glycolysis. What's the end product so far? 2 pyruvic acid. What's the fate of pyruvic acid? You don't have oxygen. If you have oxygen, I will send it to the Krebs cycle, right? You don't have oxygen. What should I do now? It depends. Are you talking about a yeast, a fungus, or are you talking about anything else? If you're talking about fungus, it's going to take that pyruvic acid and it will make ethyl alcohol out of it. And this is how they make alcohol. Did you hear about how they make alcohol? They add the yeast, did you see that before, that they have to close it, completely closed, no oxygen at all, in order to do anaerobic reaction. So what's going to happen in that barrel when they put like grape or whatever they are putting there, and they close it, right? what's going to happen? Glycolysis. And then what? No oxygen. So no Krebs cycle, no ETC. So now we have a lot of pyruvic acid, right? They add yeast to it. So the yeast is going to take that pyruvic acid, which will change it into ethanol. And they get the ethanol out. This is how they do it. If you open that barrel, no ethanol. Right? So fermentation is when you ferment, it's anaerobic. And if you do that fermentation using the yeast without the presence of oxygen, you end up having ethanol. What if if this is not yeast, if it's anything else, you're going to do lactic acid. So let me do it like this now. Pay attention. I have glucose. The cell state. Glycolysis will happen anyway. Now I have 2 pyruvic acid. What's going to have to happen to the pyruvic acid? It depends. You have oxygen, send it to the mitochondria, Krebs cycle, etc. And you end up breaking down all the way to water and carbon dioxide. All the way down. You got everything out of it. The other scenario is, I don't have oxygen. You do glycolysis anyway. What's the fate of the pyruvic acid? Mitochondria will not take it. So what's going to happen to it? It depends. Are you talking about yeast, or are you talking about anything else? Yeast, it will take that into, split it and change it into ethanol or ethyl alcohol. If that's anything else, it will change it to lactic acid. Clear so far? So here it is. Glucose, you do glycolysis, you have pyruvic acid. Is that aerobic or anaerobic? Look at this. This is the mitochondria. If it is aerobic, the mitochondria will say, okay, I will take it. I have oxygen. And it's going to change it into acetyl-CoA and send it to the Krebs cycle followed by electron transport chain. So look at this now. You took pyruvic acid to the mitochondria. Do you have oxygen or not? Yes. This is the only way to do it. You take it to the mitochondria, right? What's the fate of pyruvic acid? I will send it to Krebs cycle and electron transport chain. Is that clear? Before doing that, there is one step, intermediate step, that you change it to acetyl-CoA and then Krebs and electron transport. Is that clear? So what's acetyl-CoA? It's an intermediate step. Because you cannot simply, Krebs cycle does not utilize pyruvic acid as it is. It needs acetyl-CoA. Okay? Is that clear so far? Otherwise, as you see here, if it's a yeast, ethanol or ethyl alcohol. If it is animal cells, you change it to lactic acid. Lactate is the same as lactic acid. So here are the steps again: glucose, pyruvic acid, fate, depends. Do you have oxygen or not? Again, pyruvic acid, should I change it to acetyl-CoA and send it to Krebs cycle and then ETC or not? If not, it's fermentation in yeast, which is alcohol. This is how they make it. Otherwise, what will happen to the pyruvic acid if you don't have like an animal cells or human cells? I'm going to change it to lactic acid. And actually, it can be also used to make other things out of it. This is another fate. So normally, it will change to lactic acid, okay? But you can also use it to make anything out of it. Fat, amino acids, proteins, and so on. So if this is aerobic, what are the 3 reactions that you can do? Glycolysis, Krebs cycle, ETC, electron transport chain. Glycolysis, 6 carbon molecule will be split into 2 pyruvic acid, which is 3 carbon molecule. Okay? The numbers here are important to remember, and I'm telling you. Out of glycolysis, you're breaking down glucose into 2 pyruvic acid, isn't it? Mm-hmm. Why are you doing that? To get energy out of it, right? How many energy? How many ATP? 2. But let me say something here that's important to remember. You're not really making 2. You're making 4, and you consume 2 out of it. So the 2 is not what you're producing. The 2 is the net. You have to use 2 out of the 4. Is that clear? So in order for glycolysis to go, I'm going to use 2 and produce 4. So the net is 2. But still remember that you use the other 2 in order to run this glycolysis. Now you have pyruvic acid. I'm talking about aerobic. Does the mitochondria work or not? It will work. You have oxygen. So it will send it to the TCA, sending pyruvic acid itself. Does pyruvic acid go to the Krebs cycle or TCA? No, you have to change it to something first. Acetyl-CoA, yes. So change it to acetyl-CoA as an intermediate step. So there is a step here, right? In between this and this. Are we clear? Then you go to the TCA. TCA, tricarboxylic acid cycle. You're going to change the acetyl-CoA and you're going to break it down until you end up having the end products. What are the end products? Carbon dioxide and water. And then what? And then carbon dioxide go to the blood and you breathe it out. Isn't that how we breathe? Why are you breathing? Are you breathing to get rid of carbon dioxide and gain oxygen? So the question, where did I get that carbon dioxide from? From the Krebs cycle. Clear? So the end product here will be carbon dioxide and water. Where did you get that water from? I took the hydrogen into the oxygen. Who is carrying this electron for you and hydrogen? NAD, FAD, and the third one that's called NADP. It's not much different. NAD, H, FAD, H2, and the third one is called NADP. It's the same thing but just phosphate added to it, like the cousin of NAD phosphate. You have 3 carriers, not just 2. But these are the most commonly used, NADH and FADH2. Okay? TCA will happen in the mitochondria. This is in eukaryotes. It is an exception. Where do you make the TCA again? In the mitochondria. Where do you do the electron transport chain? In the mitochondria. Here is the question. Does the bacteria have mitochondria? No. Remember the bacteria, the prokaryotes? They do not have what? Nuclear membrane, and they do not have membranous organisms. Remember, that was a question. Remember? It does not have nuclear membrane, it does not have membranous organisms. Membranous organisms like what? Like mitochondria. So you don't have mitochondria. How can you do it in the mitochondria if you don't have mitochondria? So where does it happen? In bacteria, as an exception, in a cytoplasm. Why didn't you make it in a mitochondria? I don't have mitochondria. Right? Is that clear? Where do you make it? And out of the TCA, you make 2 ATPs. So how many from glycolysis? 2. How many from TCA? 2. Electron transport chain is going to give you 34. These numbers are important. So what's the total? 38. How many ATPs do you get in aerobic? 38. How many do you get in anaerobic? 2. Did you get this point? 2. So electron transport chain, all what's going to happen in electron transport chain is, it goes exactly like this. This is the cell, okay? This is the mitochondria, right here. Glycolysis happen here, right? And then TCA and ETC, where does it happen? Except for the mitochondria. In the bacteria, it happens in the cytoplasm. Anything else? In eukaryotes, it will happen in the mitochondria. What do you mean electron transport chain? Look at this. You take the electron with the hydrogen out of the mitochondria, and then N. And you take it out. N. Out. N. You're taking hydrogen out and N, out and N, out and N. Why are you doing that? To create a difference between the outside and inside. At the end, you'll allow to get N to get energy out of it. So what are you pumping out? Hydrogen. Hydrogen. Pumping out hydrogen. and then you're allowed to get out in, and this is the chain that's happening with the electron and hydrogen. While doing that, electron transport chain, out in, you are going to do this. And can you tell me what is this? ADP plus P, giving you ATP. What do you call this? Phosphorylation. Which one? Photophosphorylation or oxidative phosphorylation? Oxidative phosphorylation. So you're doing in this electron transport chain, oxidative phosphorylation by hydrogen bond. How about the TCA and glycolysis? How did you make the ATP? Oxidation. It's different. Oxidation, what do you mean by oxidation? I mean you take the electron by fat NAD to the oxygen. Okay? So this is aerobic, all three reactions. And here is a Krebs cycle. Krebs cycle, it's a cycle. Here's what we need to know about Krebs cycle. What are you going to use for the Krebs cycle? Pyruvic acid or acetyl-CoA? Acetyl-CoA. You cannot use pyruvic acid. You have to change it first. Clear? Okay, it's an intermediate step. So you make acetyl-CoA. And acetyl-CoA is going to go into the Krebs cycle. You put two acetyl-CoA together. Acetyl-CoA plus oxaloacetate. By the way, the Krebs cycle is a long cycle. You don't have to know anything about it except for this. Here is a TCA. Look, this is a TCA like this. I'm going to just tell you what you need to know. It's a big cycle like this. But here's what we need to know. What I'm putting in here. What should I use? Acetyl-CoA. Is that clear? And oxaloacetate, OA, oxaloacetate. That will make citric acid cycle, citric acid. That's it. There are other steps. Citric acid will change until you end up having oxaloacetate again. This is not our issue. Our issue only the first step. That's the only thing that we need to remember. Pyruvic acid change into acetyl-CoA. Is that clear? And you have oxaloacetate. Put them together to make citric acid. And this is where the name citric acid cycle came from. I want you to remember the first step only, which is when you make citric acid. How did you make citric acid? You give me acetyl-CoA, I add oxaloacetate. You have to remember everything that I'm talking about right now. Okay? So I have glycolysis. I ended up having pyruvic acid, right? I'm going to change the acetyl-CoA, send it to the Krebs cycle. What's the first step of the Krebs cycle? I'm going to add that acetyl-CoA with the oxaloacetate that I have. You end up having citric acid. That's why we call it citric acid cycle. What's going to happen to citric acid? A lot of reactions, and this is not our issue here. It will go step by step all the way. And while doing this, you will, at the end, you'll end up producing carbon dioxide. So where do we make carbon dioxide? In that cycle. So look at this. Again, I'm not talking about all the details. But as you're going with the citric acid, you're kicking out CO2. Is that clear? You end up having oxaloacetate at the end. Yes.
Okay, so then after the glycolysis, we have the acetylcholine, and then from the pyruvic acid that we created from glycolysis, right?
You create pyruvic acid from glycolysis, you change it to acetyl-CoA. Okay?
Yeah.
And then you have oxaloacetate ready for you. Oxaloacetate.
Okay.
It's something that's available in the cell.
Okay.
Another compound. You put them together, acetyl-CoA plus oxaloacetate. I put them together to make citric acid. And then lose carbon dioxide, lose carbon dioxide, lose carbon dioxide. You end up having the oxaloacetate again. Waiting for another acetyl-CoA to come. Condense. Make citric acid. Lose carbon dioxide, you end up having acetyl-CoA again. Waiting for, oxaloacetate. Waiting for another acetyl-CoA. Is that cycle clear? There are a lot of steps, but this is what you have to know. Okay? So you're producing carbon dioxide from this one step. most NADH is generated from this step, from the Krebs cycle. Look at this. You can take a picture of this, whatever you want. This is what you have to know. Acetyl-CoA, oxaloacetate, citric acid, produce carbon dioxide and produce NADH. And then you go back to oxaloacetate. Okay? But this is when you produce carbon dioxide, and this is the vast majority, not the only. Not the only. The major one that you make NADH. Okay? And you also make FADH2. But the main is NADH2. Okay? So for Krebs cycle, what you end up producing specifically, yes, you need to remember the numbers. I produce 1 FADH2, 1 ATP free, and 3 NADH. Are we clear? What is going to happen to the NADH and FADH2? They carried the electron and hydrogen. They will take it to oxygen, making water. Krebs cycle, still. So what are the end products of Krebs cycle? Carbon dioxide, water, NADH, FADH2. Is that clear? If anything is not clear, tell me. Are we clear so far? Okay, so you end up producing 1 FAD, 1 ATP, 3 NADH. Okay, what's going to happen to those? FADH and NADH and NADH is going to go to oxygen, giving them the electron and the hydrogen. It will become water, and you're producing energy. How many energy are you getting? NADH give you 3, and FADH2 will give you 2 ATPs. You need to remember this. NADH, when you give the hydrogen to oxygen, how many ATPs are generating? 3 out of NAD and 2 out of FAD. And as I mentioned before, it happened in the mitochondria, in eukaryotes, not in bacteria. And then electron transport chain. This is a chain where the vast majority of ATP is generated in electron transport chain. Can you tell me how many ATPs? 34. 34. So what is out of the 3 cycles? Which one give you most of the ATPs? Electron transport chain. And which one give you most of the carbon dioxide? Krebs cycle. Okay, so you're following. How many ATPs do you get out of NADH? 3. How many do you get out of FADH2? 2. The FAD give you 2. The NAD give you 3.
Is it 4 total though, because you have 2 acetyl-CoA for each term? It's 2, so 4 total.
4 total of what?
Oh, like CO2 or like 2 total.
Yeah, it will be doubled because of the 2, but what you need to remember is how many ATPs are generated from each one of those carriers. You as NADH, you're carrying electrons. How many ATPs are you going to end up giving me? I give you 3. You as a FAD, you're carrying electron and hydrogen. You're going to take them to oxygen. How many ATPs are you going to generate? 2. For electron transport chain, this is generally speaking for any time NADH or FADH2 is carrying. It's not only here. Generally speaking, any NADH that's carrying electrons and hydrogen, it's going to end up giving you 3 ATPs. FAD will give you 2. Here or anywhere else, it doesn't matter. This is how many ATPs can it give you. At the end, you end up adding those electrons and hydrogen to oxygen, and you will end up making water. So the end product of the 3 reactions that are the aerobic is water, carbon dioxide, and energy. Okay? This is in eukaryotes using the mitochondria. This is in prokaryotes. Where does it happen? In the cytoplasm. Okay? So here it is again, one more time. Glycolysis, pyruvic acid, 2, citric acid cycle 2, oxidative phosphorylation, which is, oxidative phosphorylation come from what? From which step? Electron transport chain. That will give you 34. Can you tell me the answer for this? Yes. If that is an anaerobic respiration, obviously anaerobic, you don't have oxygen. Which reaction is going to happen? It will be fermentation, but which of the three reactions? Glycolysis, right? Glycolysis followed by Krebs cycle and electron transport chain? No. Glycolysis followed by fermentation, either lactic acid or ethanol, right? And if that's the case, what is the final electron acceptor? What was the final electron acceptor in aerobic? Oxygen. In this one, it's not oxygen. It can be nitrate or nitrite. Nitrate is 3, nitrite is 2. NO3, NO2. Did you get the end carrier or the end receiver or acceptor? It's oxygen in aerobic. It is nitrate or nitrite in anaerobic. So fermentation, is that aerobic or anaerobic? Fermentation. It's anaerobic, right? Fermentation is anaerobic. Remember how you make ethanol? You close the barrel. Anaerobic, right? Are you going to get lactic acid or ethanol? It depends on, are you talking about yeast or animal? If this is an animal, you make which one? Lactic acid. If that was a yeast, ethanol. And it gives you a little bit of ATP, unlike the aerobic one. So this is a whole comparison between aerobic and anaerobic versus fermentation. Glycolysis, Krebs cycle, electron transport chain, fermentation is glycolysis only. The final acceptor is oxygen. In fermentation, it's organic molecule. It's in anaerobic, it is NO3, for example, and others, and so on. You can read this. This is just a summary. This is a video that's summarizing this. If you do you want to watch it now, or you can watch it later? You want to watch it? Okay, so yes.
So in the video they said from 32 to 36, but probably in the book they're saying up to 38.
Yeah,
So what is
it depends on, there are extra reactions that they didn't talk about, but we're going to stick with what we have, which is 38.
Okay.
Don't worry about this, because there is another reaction in the middle that they didn't mention that will give you the difference, and depending on if it's going to happen or not, normally it will happen, so it will end up being 38. So let's stay on the 38, which is what you have in the slides. So photosynthesis, we talked about those. If this is a plant, or even some bacteria will have the chloroplast versus the mitochondria. Chloroplast using which energy? Sunlight? Sunlight. Mitochondria, so this is photophosphorylation or oxidative phosphorylation? Photophosphorylation. And the mitochondria is going to do oxidative phosphorylation. Okay. So if this is a photosynthesis, what's going to happen? The ultimate source of all chemical energy come from sun, right? What if you don't have sun? No plants. If no plants, no animals. If no plants and animals, no humans, right? So it's always starts from the sun. And here's what's going to happen. It is a plant. Carbon dioxide, they take it from the atmosphere, right? The water, don't you water the plants? Or the plants get the water somehow? So it's going to take carbon dioxide and water and energy to give you that carbohydrate. And then when you take the carbohydrate, you break it down and use it. Okay? And remember, it's not only plants that do the photosynthesis. Do you remember the cyanobacteria and the algae? They do the same thing. Any organism that have the chloroplast will do photosynthesis. Mainly plants, but also algae and cyanobacteria. Some reactions for the photosynthesis will be light-dependent reaction, and the other one are light-independent reaction. Part of it will happen in the presence of light, and the other part will happen in the absence of light. Okay. The main job of chloroplast pigment during photosynthesis is it just capture the photons, the photons of light. Okay, that's it for this chapter.